So if you're year 13 and you're learning about the Calvin cycle, the light independent stage of photosynthesis, this might be helpful. So it's important to understand that Calvin and his co-workers actually worked this out because what they did is they took some green algae. Green algae, let's remind ourselves that that is to do with our photosynthetic pigment, chlorophyll A. They illuminated it and therefore there was light energy that was going to be absorbed. So if there is light energy going to be absorbed, that therefore means that the products of the light dependent reaction is going to be occurring. Now, in the presence of radioactive carbon dioxide, so they used radioactive carbon dioxide. So in this case, I've colored it in yellow to identify that this is radioactive. So we are trying to see what happens to this radioactive carbon dioxide. And what they did is they removed samples of the suspension at different times after they had added this radioactive carbon dioxide. Now we know in the light dependent reaction that we're going to produce our ATP and our reduced NADP. We know that in our light independent reaction, what we're going to do is, first of all, we need to fix carbon dioxide to RUBP. Well, what they did is they realized this by, after 1 second, after they'd introduced this radioactive carbon dioxide, they took the sample and the only place where they located that radioactive carbon dioxide was in GP. So the fact that it was only found in GP showed that the carbon dioxide had to be fixed into the GP. So the RUBP and the radioactive carbon dioxide were combined to now give us our GP. Now they continued to do this, and after 5 seconds, when they removed that sample of the suspension, what they identified is after 5 seconds, this radioactive carbon dioxide was not only located in the GP, it was also found in TP, it was found in some of the glucose phosphates, and it was also found in some of the RUBP, therefore showing that the TP had to regenerate into RUBP, and also showing that the GP had to be converted into TP. They continued to do this after 30 seconds, and what they noticed is there was more molecules which all contained this radioactive carbon dioxide. So therefore what they were showing is that this was a cycle, and this cycle was always going in the same way, where you would have your RUBP fixing the carbon dioxide into the RUBP, which create our GP. Our GP would then be converted into our TP, and then that TP would either become any of the other molecules or regenerated back to the RUBP. So this is the evidence that proved, and this is why it's now called the Calvin cycle.